Hello everyone, welcome to the channel. If you're new here, I'm Zach Hancock. I'm an evolutionary biologist who specializes in population genetics, phylogenetics, and genome evolution. I'm a postdoctoral researcher at the University of Michigan, where I develop statistical techniques to analyze genetic data. On this channel, you'll find a mix of evolution education, the history and philosophy of science, and some pseudoscience debunking. Today we're going to talk about one of my favorite topics, the evolution of genome complexity. Even if you have little or no background in genetics or molecular biology, you've undoubtedly heard that genomes are incredibly complex molecules with extensive interacting networks and 3D structures that produce even more complicated products, such as enzymes that are composed of dozens of interacting proteins, all linked together to form a necessary functional unit. Indeed, in many of these final protein products, all of these subunits are necessary to ensure the proper function of the protein, given the appearance that these molecules are irreducibly complex, to borrow a term from the biochemist and intelligent design proponent Michael Behe. You've probably also heard the prevailing narrative in popular culture that evolution of molecular complexity is due to slight modifications over time that each contribute in a stepwise fashion to increase the fitness of the organism, either by improving the fidelity or catalytic properties of the individual proteins involved, or perhaps by diversifying the regulatory apparatus. And indeed, this narrative is downright Darwinian. Evolution proceeds by slight modifications that increase the fitness of the organism, and further that complexity itself is a reflection of Darwinian natural selection. In many ways, this line of thinking is reminiscent of Aristotle's scala natura, or the scale of nature, in which increasingly complex organisms are considered higher. Animals above plants, plants above microbes, etc., with man, of course, on top. Few serious biologists would agree that this ladder of progress is technically correct, despite falling prey to its central assumption that something about complexity is inherently indicative of progress and must emerge by a positive Darwinian selection. In this video, I'm going to explain how genomes and their products can evolve complexity, but instead of relying on the simplistic narrative that complexity is solely driven by many rare beneficial mutations selected over time, we're going to consider the actual features of genomes, their biochemical properties and architecture, and we're going to juxtapose these with expectations from theoretical population genetics. In doing so, we'll see that natural selection is actually quite conservative, not creative, and in general acts to preserve simplicity and against complexity. That complexity is actually the result of effectively neutral processes and mutational bias, particularly towards deleterious, function-disrupting mutations, and that once complexity arises, selection acts to prevent further degradation of function. But before we can dive into this, we really need to discuss what is complexity. There are several ways we can think about this. Generally, complexity is a measure of the number of parts of a system and the degree of their interactions or interdependencies. If we think about a network of interacting nodes, a simple network would be composed of fewer nodes with fewer interactions between them than a complex one that may have many interacting parts and extensive interactions between them. We also generally think of these interactions between parts as necessary to some extent to the overall function of the system. Again, there's a dependency between the system as a whole and the underlying network that dictates the nature of the system. So let's think about this on a genomic level via a comparison between prokaryotes and eukaryotes. Comparatively, prokaryotes have much simpler genomes. Much of their genome is devoted to producing protein products. Very little of it is intergenic. They have overlapping reading frames that enable multiple proteins to be regulated and transcribed under the control of a single promoter. Hence, they have few regulatory elements that are themselves quite compact. Prokaryotes have no or very few introns, and hence no need for spliceosomes, and they have very few transposons or repetitive DNA. Their genomes are rarely greater than 10 megabases and typically much smaller. Even the protein synthesis machinery, the ribosome, is simpler with fewer small and large subunits than what is seen in eukaryotes. Finally, compared to eukaryotes, prokaryotes have very large population sizes, on the order of billions or even trillions of individual cells. Eukaryotes are much more complex. The genome is bound within a nucleus and the coding region is in pieces, each exon sandwiched between long stretches of introns, regions that must be spliced out after transcription but before translation. They have extensive regulatory networks that can be thousands of bases upstream of the reading frame. Many of their proteins are more elaborate as well, with dozens of proteins coded from different regions all interacting to form large macromolecules dedicated to some singular function. Eukaryotes also have expansive transposon families that in some species occupy as much as 90% of their genome. In humans, at least 50% is occupied by defunct transposons that are no longer capable of transpositioning due to a combination of gene silencing and mutational processes that have degraded them. Lastly, eukaryotes have typically much smaller population sizes, even among the unicellular eukaryotes, with the upper sizes being in the hundreds of millions, but many large-bodied eukaryotes, such as vertebrates and land plants, being in the tens or hundreds of thousands, orders of magnitude smaller than prokaryotes. When we look at scaling relationships between genome size and genomic content, we find interesting patterns across the tree of life. Prokaryotes, as I mentioned earlier, have large portions of their genome dedicated to proteins, but as we increase in complexity into unicellular eukaryotes and eventually vertebrates and land plants, we see that the proportion of the genome devoted to coding regions actually plateaus. Indeed, in most large eukaryotes, less than 1% of the genome is exonic. So what is expanding then? Above 10 megabases, we see a stark increase in intergenic DNA, especially transposons, introns, and pseudogenes. Hence, this added complexity in eukaryotic genomes is not coming from coding elements, but from expansions of non-coding elements. Now that we have that background out of the way, we can try to understand how we go from something simple, like a prokaryotic genome, to something more complex, like a eukaryotic genome. To begin, let's first take note of a serious problem with the typical Darwinian explanation of the origin of complexity. To grasp the ubiquity of this problem, we need to refresh ourselves on some key concepts from population genetic theory. There are three mechanisms we need to consider: natural selection, genetic drift, and mutation. The first two act to remove variation from populations by either fixing or driving them to extinction, while the third is constantly adding new variation every generation. Since natural selection and genetic drift are both acting to change the frequencies of existing variants, how do they interact with each other? Recall that selection is merely the change in the frequency of any allele due to its contribution to fitness. Fitness in an evolutionary context is measured as the number of offspring you have relative to the population average. So any allele that increases your fitness increases in frequency and expectation, while any that decreases your fitness will eventually be lost. That change in frequency is what we call natural selection. Genetic drift doesn't care what the allele does. Every generation, due to a combination of Mendelian inheritance, the chance passing of only one of your genomes to the next generation, as well as chance sterility, you died from something outside of the control of your genes, and hence you had no children, allele frequencies will change. This effect is what we call sampling error, and it derives from the fact that all populations are finite, and hence the next generation won't display the exact allelic frequencies of the preceding. By this sampling effect alone, some alleles are lost and some are fixed, irrespective of what they do. The strength of this sampling effect is dictated by population size. The smaller the population, the more likely chance is going to play a role in changing frequencies. In a simple case, you can imagine tossing a coin 10 times. We know that the probability of getting heads or tails is 50%, but by only flipping 10 times, you may find you get heads 80% of the time and tails 20%. By chance, you've deviated from expectation. Flip it 10,000 times and you'll be much closer to that 50-50 expectation. Thus, by increasing your sample size, or population size in a biological context, you've decreased stochasticity. We should see that natural selection and genetic drift are opposing forces. Selection is deterministic, based on the contribution of an allele to fitness, while drift is random, irrespective of an allele's effect on fitness. How can we determine which of these two forces will win out? Intuitively, the winner in this tug of war is determined by the relative strength of selection to drift, which we can express as the absolute value of S is greater than or less than 1 over N, where S is the selection coefficient favoring or disfavoring an allele, and N is population size. When these values are equivalent, or when 1 over N is greater than S, drift dominates the frequency of the variant, irrespective of if S is technically positive or negative. We call this effective neutrality. We can write the probability of fixation of an allele of any selective effect as follows. Plotting this, we see that as the strength of selection deviates from 0, the shape of the probability of fixation curve is strongly influenced by the population size, with smaller populations having a much higher probability of fixation for even deleterious alleles relative to those of much larger size. Further, when selection is 0, indicating neutrality, the probability of fixation is equal to 1 over N, or the starting frequency of the new mutation. Now that we understand the interaction between selection and drift, let's discuss mutational processes. Empirical studies have found that mutations are overwhelmingly deleterious, even if only slightly. Indeed, it's possible that there are very few completely neutral mutations such that S equals 0. Likely, nearly all have some selective disadvantage, even if vanishingly small. Why are most mutations deleterious? Well, imagine a gene duplication, for example. This is a mutational event that hasn't disrupted genic function, but it has added a redundant copy to the genome. This redundant copy must be replicated, which costs cellular energy in the form of ATP. This dedication of ATP to this new region comes at the expense of the total cell budget of ATP, making it a costly investment, given that the gene copy is redundant. Duplications can also disrupt dosage, adding more protein products than is necessary for cell function and stripping resources from other proteins. Point mutations, though less costly, can still technically involve a cost. For example, if a third codon position mutates such that a new codon is coded for, even if the same amino acid is specified, if there are fewer tRNAs to carry this new codon, translation efficiency is reduced. Again, these effects can collectively be very small, but they still technically cost the cell resources and hence, strictly speaking, are deleterious. With that being said, there are beneficial mutations that have been measured empirically. In studies on experimental evolution, we find that extensive examples of beneficial mutations arising that increase cell growth rates, the ability of cells to grow on different mediums, etc. We also find examples of compensatory mutations, those that emerge and repair previous deleterious mutations that disrupted function. However, these are exceptionally rare, and if we want to construct a general model of the evolution of complexity, it seems prudent we not rely on the rarest of events. Thus, from here on out, we won't consider beneficial mutations at all. We are going to proceed as if all mutations are either deleterious or strictly neutral. Now that we've primed our expectation via population genetic theory, let's return to the two genomes, the simple prokaryote and the complex eukaryote. Let's entertain the Darwinian perspective for a moment, that complexity is derived from natural selection. In which of these two should natural selection be the most efficient to drive complexity? Recalling that prokaryotes have population sizes on the orders of billions or even trillions, and that from population genetic theory, selection is most efficient in organisms with very large population sizes because the power of drift is reduced, we should expect to see the greatest complexity in prokaryotes. Of course, that's not what we see at all. Prokaryotes possess the simplest genomes, not the most complex. Well, what gives? Remember from our discussion of mutations above that most mutations are deleterious. Hence, selection is rarely acting to drive a mutation to fixation. Mostly, it is purging them from populations. That is, selection is a highly conservative force. This should tell us something key about evolving complexity. Remember from our definition of complexity that what is required is many nodes in a network all interacting with each other. We need to build up our network of interactions from simple beginnings. Well, how does that happen at a genomic level? To increase node number, and hence the potentiality for interactions between nodes, we need to increase the amount of genomic content in the cell. This is achieved via duplications and insertional events. But remember that these events, at least initially, have no functional benefit, and in fact can be costly to the cell, even if only slightly. For these networks to increase in complexity, they need time for mutational processes to drive divergence between duplicates. But if selection is constantly removing them, such divergence can never accrue. So, what are the fundamental conditions necessary to evolve genomic complexity? First, we need a selectively permissive environment. We need mutational processes, even deleterious ones, to have the opportunity to add things to the genome. If selection is constantly purging things, complexity is stifled and efficiency and simplicity will remain, as in the case of prokaryotic genomes. Another consideration is that complexity is hazardous in and of itself. As a network of interacting parts gets larger and larger, you increase the number of ways to break the system. Hence, it becomes progressively easier to render the system non-functional the more parts it relies upon. These two notions form the foundation of the mutation hazard hypothesis, put forward by Michael Lynch. It is one of the two key theories for explaining the evolution of genomic complexity, so it's worth explaining it here. In short, it posits that most mutations are deleterious, and that selection should act to remove mutations and keep molecular machines at the highest level of efficiency. Simplistically, we can think about it as selection is trying to drive molecular perfection. However, drift is opposing this. It allows deleterious mutations that are preventing molecular perfection from being attained to reach high frequency and even fix in populations. Hence, there is always a gap between absolute molecular perfection and any existing molecule. And the distance of this gap grows wider as drift becomes stronger due to selection's decreased efficiency at purging deleterious mutants. The mutation hazard hypothesis also makes strong predictions about the evolution of the mutation rate itself. Since mutations are overwhelmingly deleterious, selection should act to increase the efficiency of DNA polymerase replication fidelity, as well as its proofreading mechanisms, and should improve the action of mismatch repair enzymes. Hence, organisms with larger population sizes are expected to have lower mutation rates than those with smaller sizes due to selection being inefficient at smaller sizes to prevent mutation accumulation in DNA polymerase and mismatch repair enzymes. And indeed, that pattern is exactly what we see. Prokaryotes have the lowest per generation mutation rate of around 10 to the negative 11, while large-bodied eukaryotes have very high mutation rates. Humans, in fact, have the highest on record at 10 to the negative 8, reflecting our very small population sizes over much of our evolutionary history. It should be pretty clear by this point that my argument is that genomic complexity emerges not from Darwinian selection, but by its absence. That only in selectively permissive environments can mutational processes coupled with genetic drift be allowed to increase complexity. Let's take a simple case. A gene undergoes a duplication. This mutation increases complexity because it increases the number of nodes in a system. Further mutational processes, since they are mostly deleterious, degrade the function of each gene, both the parent and the duplicate, because the original copy is freed from purifying selection since the duplicate can now compensate for any deleterious mutation. Over time, we go from a situation in which one gene was capable of carrying out the necessary function to both genes being required, as each have mutated such that neither alone can function anymore. Hence, we built an interaction between these two genes, further increasing genomic complexity. If we were to examine this system after the fact, we'd come to the conclusion that this gene network is irreducibly complex. You can't have one without the other. We call this process subfunctionalization, and it's one of the prime ways of increasing genomic complexity via initial redundancy and subsequent degradation of redundant copies. Again, such redundancy wouldn't be allowed to persist if selection were too strong. It would immediately be removed due to the increased cost of the cell. But in organisms with smaller population sizes such that mutation is effectively neutral, it can persist long enough and subsequent mutations can disrupt individual function. This idea of the evolution of complexity falls under what is known as constructive neutral evolution, or CNE. This hypothesis reverses the normal Darwinian order of events, that a simple molecule becomes more complex by slight beneficial variance, and that fitness is always increasing. Instead, CNE claims that simple molecules become complex by a combination of excess capacity, genetic drift, epistasis, and subsequent purifying selection. Let's briefly explain how each of these interact with one another, and then we'll give some specific examples of complex molecules that evolved under this framework. The concept of excess capacity refers to the generation of some product above what is necessary to maintain normal cell function. A prime example of this is gene duplication. Having two copies of the same gene doubles the gene products, leading to an excess. This excess permits some of these products to interact epistatically with other protein products in ways that don't reduce the efficiency of the original product. In doing so, they enable many mutations that would be deleterious in the absence of this epistatic interaction to become conditionally neutral. That is, this interaction permits loss of function mutations to occur because the interaction itself buffers the original product against the deleterious mutant. This interaction becomes cemented and necessary once these mutations accumulate to such an extent that in the absence of the epistatic interaction, the individual products no longer function. Hence, any mutation that would break the interaction is under purifying selection, which then stabilizes the relationship. To summarize what we've discussed so far, for genomic complexity to evolve, you need a selectively permissive environment such that genetic drift can dictate the frequency of mutations that may otherwise be deleterious. You need there to be an excess capacity of gene products to permit some of them to interact in novel ways without disrupting the original purpose of the gene. Again, this is accomplished via gene duplications that increase the product of a gene beyond what is necessary for cell functioning. You then need those epistatic interactions to become required for function to be maintained in the face of independent mutational processes, breaking the function at the original gene level. Finally, purifying selection maintains that interaction against anything that would disrupt it, because it would lead to a complete loss of function. It's important to note here that these interactions are purely biochemical. There's nothing special about them. If you mix any collection of molecules in an aqueous solution, they will bind in predictable configurations given their underlying valence electron distributions. Nothing has to facilitate these interactions. The same is true in the cell, which is effectively just a closed aqueous solution. Post-protein synthesis, many will be picked up by chaperone proteins and shuttled to particular parts of the cell, while others, in the case of excess capacity, may be free to bind in novel ways with existing proteins in the cytoplasm. These bindings, again, are no different than how inorganic molecules interact in a beaker. They follow the basic laws of chemistry. So what are some complex genomic features that can be explained under this framework? One that occupies a considerable portion of all eukaryotic genomes are introns. Some introns are capable of self-splicing. That is, they don't require the big complex spliceosomes. These are the group 1 and group 2 introns. The former catalyze their own reaction via the attachment of an intron cofactor, most commonly a free guanosine, followed by transesterification, which results in a free linear spliced RNA intron. Group 2 introns utilize a free reactive adenosine with a 2' hydroxyl that will bind to the 5' phosphate of the upstream guanosine, forming a loop and then cleaving it from the exon. This freeing up of the first exon means there's a free hydroxyl group at the end of the first exon, which then attacks the phosphate group at the end of the intron loop. This cleaves the intron off, leaving it in this kind of necklace structure, fittingly called a lariat, and ultimately joins the first and second introns together. Okay, so some introns can self-splice, so why do we need spliceosomal proteins then? One, we should note that spliceosomes are composed of many proteins bound around small nuclear RNAs, and these small nuclear RNAs are pieced together to resemble the same splicing machinery of group 2 introns. In fact, the splicing machinery of spliceosome complexes is the exact same as group 2 intron self-splicing. Hence, this machinery could evolve first by a separation of the individual catalytic components of group 2 introns via duplications, which then would produce the individual small nuclear RNA components. Various experimental studies have found that separated intron catalytic components will spontaneously rejoin. The existence of these components permit group 1 and group 2 introns to then lose their self-splicing capacity, since there now exist small nuclear RNAs capable of picking up the slack. That is, these introns were freed from the selective pressure to maintain self-splicing function. Next, any proteins that bind to these small RNAs that stabilize their structure could enable additional mutations to degrade their individual structural integrity, thus further forcing these complexes to form to ensure proper intron splicing via purifying selection. In this way, a kind of accidental dependency arises via a combination of mutational processes and adventitious RNA protein binding. Once the machinery is in place to splice out the introns incapable of self-splicing, introns are then freed from selective constraint and are permitted to expand. This expansion is limited only by the availability of spliceosomal complexes and the selective efficiency relative to genetic drift. Each added intron increases the cost to the cell, and this cost cannot exceed what the cell can tolerate in terms of survival, or what it will tolerate in terms of selective efficiency. So long as the proliferation does not exceed the cost noticeable by selection, introns will persist. Notice how we've now seen a scenario in which genomic complexity in the form of introns and the spliceosome complex can emerge entirely in the absence of positive Darwinian selection, and indeed may only have emerged in the absence of positive Darwinian selection. But where did introns come from in the first place? Recent work has found that introns strongly resemble many known DNA transposons, and that the self-splicing activity of group 1 and group 2 introns indicate that they most likely emerged first as mobile genetic elements, that is, genomic parasites. Hence, the evolution of genomic complexity with regards to the patterns of eukaryotic genes, which are in pieces due to intron sequences, was driven by molecular accommodation, drift, and mutational processes. Another great example of constructive neutral evolution is the ribosome. Ribosomes are gigantic, complex molecules composed of dozens of proteins that exist as individual subunits, both large and small, that may have thousands of copies across the genome. In addition, ribosomes are protein RNA complexes, with the RNA loops forming the important binding site for the tRNAs that are transferring mRNA to the ribosome for translation. Each of these protein compartments need to be in place for this big macromolecule to function. How in the world could it have possibly evolved? Isn't this a perfect example of irreducible complexity? A few cracks in this logic emerge immediately. First, the ribosomal subunit number and composition is not conserved across lineages. Many organisms have different numbers and in different compositions, despite all of them functioning exactly the same. In fact, prokaryotes have much fewer ribosomal subunits than eukaryotes, despite there being no difference in the function of the ribosome. In addition to the expansion of protein subunits, the size of the ribosomal RNAs have dramatically expanded as well in eukaryotes, especially large-bodied eukaryotes compared to prokaryotes. Many studies have suggested the origin of ribosomes was merely a ribozyme, the ribosomal RNA components of the modern ribosome. If so, protein subunits could have bound adventitiously to this initial ribozyme. If mutations then accumulated in the genes for this ribosomal RNA that degraded stability, these bound subunits may have themselves acted as stabilizing factors. Subsequent duplication of these protein subunits could have dramatically increased the binding affinity of these incipient ribosomes, which has empirical support first from the large number of gene copies of ribosomes in eukaryotic genomes, coupled with a highly disparate composition of protein subunits of the otherwise core complexes across the tree of life. It's important to note here that while these arguments thus far have solely relied on mutation being a destructive force, and that all increases in complexity are being driven by chance associations of molecules, this does not mean that natural selection has played no role in the evolution of these molecules. Instead, my argument is that it has played no role in their initial evolution. Natural selection may have tinkered with them once they were together and in place, elaborating on the basic structures, but I contend would have never favored such bloated structures in the first place. The American cartoonist Rube Goldberg became a household name in the 1920s and 30s for his depictions of elaborately complex and impractical devices. Here's an example known as the self-operating napkin, in which Professor Butts has his mustache wiped for him by a ridiculous machine. A more modern example might be the game Mousetrap, in which players construct increasingly elaborate traps with the sole purpose to catch a mouse. These bloated, complex devices are reminiscent of big eukaryotic genomes. They exist as an intricate network of interacting parts, of which a fault in any could cause the whole structure to come tumbling down. I have tried to portray this complexity in a way different than what you might have heard. Instead of being a supreme product of selection, it's instead a quirky Goldbergian product of bias, mutation, and drift. That selection is acting against these Goldbergian intricacies, fighting to keep genomes streamlined and simple. In this way, prokaryotes are the pinnacle of Darwinian evolution. Their genomes are highly compact molecular machines. Eukaryotic genomes, especially those of large-bodied eukaryotes, such as vertebrates and land plants, are virtually non-Darwinian mazes of nonsense that, when looked at from afar, much like Goldberg's ridiculous devices, might appear to be composed of exquisite complexity. And indeed they are. It's just that that complexity is redundant, unnecessary, and at times completely ridiculous. The last thing I should address here is whether the most complex genomes are better in some way than the simpler ones. One might say, okay, yes, the eukaryotic ribosome is bigger and more complicated, but maybe it works better than the prokaryotic one. But there's no data to suggest that's true. In fact, prokaryotes have a slight advantage in terms of translation rate, and we're virtually equivalent with regards to translation efficiency. The same is true of spliceosomes. Group 2 introns with self-splicing mechanisms are actually more efficient at splicing than the big bloated spliceosome complexes. Further, if we just consider the basic mechanism of subfunctionalization, obviously prior to duplication and mutation degradation, you only need one gene to generate the desired product. Subfunctionalization, despite increasing complexity, decreases efficiency. Now you need two to do the same thing. So, how does genomic complexity evolve? I contend we shift the popular discourse around this topic, including among biologists, to account for both the molecular biology and population genetic theory. When we couple these, we find that complexity is rarely the result of natural selection, and hence we need to search the other mechanisms of evolution to understand its emergence. When we do so, we find the data is much better accounted for, that complexity is correlated with the reduced efficiency of selection, not the other way around. By integrating our understanding of mutational bias and the overwhelming deleterious nature of mutations, we can begin to understand the conditions that permit complexity to emerge. Finally, I think the mutation hazard hypothesis and constructive neutral evolution are far better in line with both empirical data and population genetic theory than purely adaptationist explanations for complexity. Thanks so much for being here. I know this was somewhat of a dense topic and may fly in the face of some prevailing narratives that you may have heard, but at the genomic level, a clearer picture has emerged over the past 20 years or so that mutational and stochastic processes cannot be ignored, and that Darwinian selection has been afforded too much credit as a creative as opposed to conservative force in evolution. If you have any questions, I'd be more than happy to discuss this further in the comments below. Thanks so much for being here. Like and subscribe for more content, and I'll catch you next time.